Hi, I'm Jonah, editor-in-chief at pharmaphorum. I'm here with a post-ASCO interview, virtually chatting with Matteo Levisetti, Chief Medical Officer of Q Biopharma. We're going to talk a little bit about Q, some of the data that they presented at the show now about a month ago, and just talk trends in oncology too. So, Matteo, thanks for joining me.
Thanks for having me, Jonah.
So we were meant to meet on the show floor. That there was a mix-up on my end with my flight, and I had to cancel our meeting. So I really appreciate you making the time after the fact. Tell me about what you were doing at ASCO and that some of the data that Q was presenting.
Certainly. So Q Biopharma is a clinical stage biologics company with two molecules in the clinic. And we were selected for an oral presentation at ASCO for an update on data on our head and neck cancer program. And so to date, we've treated actually 80 patients. Okay. 39 patients in the late line setting, so in the second line and beyond, with Q101 monotherapy, and then another 31 patients with Q101 in combination with pembrolizumab. And so our molecule Q101 is a T cell engager that selectively delivers IL-2 to tumor-relevant T cells. So it's a complex that includes a peptide HLA and IL-2. So it basically gives two signals to T cells that are specific for cancer cells. And it leaves all the rest of T cells in the patient, which are really irrelevant to eradicating cancer, unstimulated, unactivated. And so as such, we've achieved really remarkable tolerability and activity in terms of controlling late stage cancer and then in the frontline setting. And so in the frontline setting, we presented updated data where we've observed an overall objective response rate of 46% in treatment naive patients in the first line recurrent metastatic setting. And this is really quite notable if you compare it to historical response rates. For example, the pivotal KEYNOTE-48 trial, where the objective response rate was 19% in the frontline setting. These responses are very durable. The median duration of response has not been met. And now, as the PFS and survival matures, we've observed a 96% 12-month overall survival, and that compares very favorable to the historical data of about 55%. So the other really very interesting observation is we see responses in patients that have low PD-L1 expression. So those with CPS scores less than 20. And if you look again back at the KEYNOTE-48 trial, patients with low CPS scores actually had a response rate of 14%. And if you look at the 12 patients of the 24 that we reported on, with Q101 plus pembrolizumab, we observe a response rate of 50%. So again, it is a single-arm study, but really the data now compares very favorable to historical metrics. We're planning now to move forward with a randomized phase 2 trial comparing the combination of Q101 with pembrolizumab to pembrolizumab monotherapy.
Great. Yeah, so that's really interesting, really promising, even though it's still kind of in the early stages of the research.
Yeah, no. We're very encouraged, and again, it's really the metrics that are emerging in survival that we find perhaps the most compelling and interesting. And so, in the patient monotherapy group that we treated, we expanded at a recommended phase 2 dose of 4 mg per kg, and this is in patients that had progressed on prior platinum-based chemotherapy and checkpoint inhibitor therapy. And now we've observed a median overall survival in this group of more than 20 months, so 20.8 months. And this again is quite notable. And every year at ASCO, I would ask our investigators, well, what would be interesting? Okay. and they would say, well, it's single arm data, and then they all would come down to about, well, 12 months would be kind of notable, right? And so now we're beyond 20 months, and our investigators have really taken note. If you look at the second line pivotal trials for both nivolumab and pembro in second line head and neck cancer, the median overall survival is just about 8 months. So to observe an overall survival of more than 20 months in a later line setting, i.e. the third line setting plus, is notable. And again, if we look at the entire comprehensive data set that we have, our drug basically modulates the patient's immune system in such a way that it expands a population of tumor-specific T cells that stick around. And we feel this is the substrate that underlines this observation of really improved survival. Okay, and again, in the frontline setting, we anticipate that the survival, will really be differentiated from historical parameters as well. And it's great that we're seeing an objective response rate that's durable of close to 50%.
So head and neck is not the most kind of common area that we see data about at the show. I mean, tell me a little. I'm not as familiar with that space. Tell me a little bit about kind of what are some of the particular kind of challenges and contours there.
Well, it's a therapeutic area that's really been transformed in the last 5 years with the approval of immunotherapy. Okay, specifically checkpoint inhibitor therapy. So historically, for a couple decades, there had been little improvement in treatment options for patients with squamous cell carcinoma of the head and neck. There basically was extreme regimen, which is a chemotherapy regimen with platinum-based chemotherapy and cetuximab. And that's the standard of care, for patients that can tolerate it for the last 10, 20 years. Okay. And so it was really nivolumab that did the first trial in the second line setting. So these are patients that had progressed on prior chemo, platinum-based chemo, were then treated with nivolumab monotherapy. And there they demonstrated a survival advantage. And the same was done then with the Keynote 40 pembrolizumab second line trial. So for the first time, this became an option for patients that were chemo refractory, much better tolerated regimen with a survival benefit. And then with the Keynote 48 trial, this is now in the frontline setting. Okay. So treatment naive patients with recurrent metastatic head and neck cancer, again here, a benefit in survival was demonstrated with pembrolizumab monotherapy. Okay, so again, a very different regimen compared to extreme chemo-based therapy up front, and that really sort of revolutionized, the first-line treatment of patients with head and neck cancer. And again, pembro is approved as first line as monotherapy and in combination with chemotherapy. And so really now as standard of care, physicians, choose between those two options. If the patient has low PD-L1 expression or CPS less than 20, and the patient's in pretty good shape where they can tolerate chemo, the preferred regimen would be chemo plus pembro. And that's where, the combination of Q101 and pembrolizumab really is attractive because it's really well tolerated. And so we see nothing, the 70 to 80% grade 3 related AEs that you see with a chemo regimen, we see less than 30%, and nothing really concerning. So again, a very, very differentiated tolerability profile.
So tell me a little bit about Q Biopharma. What are kind of what's the sort of core mission of the company, and what are some of your differentiators when it comes to the technology that's underpinning this study?
Yeah. So really, the core, but what's at the center of Qube's platform and technology is really our objective and mission is to modulate the immune system in such a way to eradicate cancer. And we also have programs that are preclinical where we aim to modulate the immune system to suppress autoimmune disease or to delete autoreactive T cells. And so in the setting of cancer, we took the approach of really making the only, tumor-specific specific T cell engager. And so we have a construct that engages the T cell receptor only of T cells that are specific and react to the cancer antigens. And at the same time deliver IL-2 signaling through 4 attenuated IL-2 molecules. And so, as I'm sure you're aware, in the last 5-10 years, there's been many companies, right, that have tried to leverage the benefits of IL-2. So with the IL-2 muteins and, Nectar, Synthorix, there's been 7 or 8 that unfortunately for patients have not really borne out in large phase 3 trials. They've basically shown no benefit. And again, these are non-selective. So they deliver IL-2 to every T cell in the patient, of which 99.9% are irrelevant to cancer. So they're limited to a lesser degree than the tolerability issues and liability with wild type IL-2 proleukin. But hand in hand with that, they're not. They didn't appear, to provide any additional efficacy, in combination with checkpoints or chemo. So our platform is really differentiated in that it really selectively delivers IL-2 to the relevant T cells, and as such, we can give very high doses of IL-2 in a highly tolerated manner. And so we have a second molecule. So the first one targets HPV positive head and neck cancer. Now we have a second molecule in the clinic that we're treating patients with cancers that express WT1 or Wilms tumor 1, an oncofetal antigen. And here we're treating patients now with colon cancer, pancreatic, gastric, and ovarian cancer. And we're seeing findings that are very much reminiscent of what we've seen with 101 with regards to the tolerability, the expansion of anti-tumor T cells. And then, even in dose escalations, we're seeing significant reductions in tumor burden in patients with advanced gastric cancer, ovarian cancer. And so here our plan is to move forward with combining Q102 with a checkpoint inhibitor, anticipating that the findings may recapitulate what we saw in head and neck when we combined with pembrolizumab. And now many of these tumors, sort of typical wild type, colon, gastric, ovarian, those are not checkpoint responsive tumors. And so we're hoping that we can sort of open up, a new treatment option in some of these cancers that have been resistant to checkpoint inhibition, that, really underwhelming activity with CPIs.
That's fascinating. Yeah, I mean, I know that ASCO this year was like the 10th anniversary of Keytruda, and so the notion that even now, 10 years later, we're finding these unlocks to get even more value out of that class of drugs is fascinating. There are so many different kinds of cancers, that that you have to kind of try to figure out how to adapt these treatments to. What else did you see or hear around the conference that you found interesting? I mean, we have the advantage of having it in the rearview mirror. Either around some of the same things that you guys work in, or even other spaces that caught your eye.
Well, we've of course followed very closely related approaches with cancer vaccines that have been now explored in combination with pembrolizumab, actually, in the same population. And there, I think, and there were a couple special sessions on cancer vaccines, and I think, unfortunately, the limitations that have plagued, the success of cancer vaccines remain the same as they have for 30 years. Okay? So one is really limited by the number of vaccine administrations you can give. and by the durability of the immune response that they invoke, and the strength of that immune response as it translates into an efficacy population or an effector T cell population. If you immunize a patient, or you give antigen repeatedly, you actually create tolerance. Okay, so with Q101, we bypass all of that. We're able to directly deliver the appropriate signals to the effector T cells that will then go eradicate the cancer. Okay. So again, there are multiple, even, sort of overview summary talks, but then two or three, other companies that are working in the space where we're just not seeing the same response rates, and certainly not the durability. And no, our survival data is evolving, and we feel that it's just going to strengthen. And I think that's where, with our repeat dosing, our drug is given every 3 weeks. With the vaccine regimens, you're limited to perhaps 3, at most 4 treatments early on. And I think that for reasons that we know very well with regards to how the immune system responds to repeated vaccination, you generally do a prime boost, right? So even with early protective vaccines in kids and things, the immune system has not necessarily an idiosyncratic response to repeated challenge, but again, that's the expected outcome.
Does what it's designed to do, right? Yeah.
Yeah.
Gets better at. Yeah.
Yeah.
My understanding with those vaccines is that they are meant to work in combination. They're meant to keep cancers from coming back after you get rid of them with other treatments. So, I mean, is it an either or does these, what you're working on, potentially stack on top of that, or work with those vaccines?
Yeah, well, it's a great question, and what's remarkable in what we're observing now. So we've treated patients up to 2 years, okay, with the immunostat, the drugs we call immunostats, and so we have a few patients that have stopped treatment, okay, for various reasons. It could be an adverse event related to pembrolizumab. And we have at least 3 patients that have maintained objective responses for a long period of time, so even over a year. So we have a patient with a complete response who's been off treatment for 18 months. Okay. And so the only interpretation I have for that is that the treatment with Q101 and pembrolizumab, for 6 months, basically, resulted in basically anti-tumor immunity that's kept this patient cancer-free for a year and a half off treatment. And so it's hard to, whether it's completely sterilizing immunity, as in that all the tumor cells are gone, or if there are some, residual micrometastatic deposits of tumor, the immune system has managed to keep them under such good control that there's no clinical manifestation and there's no radiologic detection of those cancer cells, if they're even there. So again, I think as we move forward, there's certainly a potential, right, that this therapy will result in prolonged protection even after, the therapeutic intervention phase. But again, I don't want to get ahead of ourselves. It's
gotta wait and see the data, right?
We got to wait and see what the data shows us. But from an immunologic perspective, it's consistent with what we believe to be the mechanism of action. And there's, the field has learned more and more about certain T cells and T cell exhaustion, and signals that T cells need to end up being sort of viable, healthy effector T cells that can renew themselves. And so a lot of this pioneering work comes from Rafi Ahmed at Emory, But we feel that the treatment of the immunostat, giving these two signals to a T cell through the T cell receptor and an IL-2 in the presence of blocking PD-1 may very well result in the differentiation and expansion of a sort of a stem-like population that can renew itself and generate very, very healthy, good effector T cells. So not exhausted cells, but effector killer T cells.
So we're reaching the end of our time, and it's really, really interesting stuff. Any final thoughts, or what would you say is sort of the key takeaway here for folks to remember about the work that you've done here and that you're continuing to do?
Yeah, well, I mean, it's really for the first time, demonstrating, in patients, right, that one can selectively modulate and expand a very specific population of T cells that then have very meaningful anti-tumor activity in a way that's very well tolerated. Okay, and so all other applications, all other approved drugs, checkpoint inhibitors are not selective. Okay, you get not insignificant adverse events, immune-related adverse events from all checkpoint inhibitors. All T cell engagers, whether they're a CD3, CD19 BiTE, or the evolving field of CD3 with whatever target against a tumor antigen, there's very significant tolerability issues with neurologic side effects and cytokine release storms. So here, I think it's really the first example we're using a biologic construct that is made up of the peptide HLA, okay, that interacts with the T cell receptor. It is unique, and beforehand we thought, well, is there going to be immunogenicity and anti-drug antibodies? We've seen no clinically significant anti-drug antibodies. Is this really going to work? And so remarkably, it works, and it results in differentiated clinical activity early on, but that's where we start, right? And so we've also demonstrated that we can take a second molecule. Just the only difference there was swapping out the peptide on the HLA, so it expands a different group of specific T cells. All the rest is identical, 99% identical. So that's a substrate to put in many different epitope targets and target many different tumors by modulating that patient's T cell repertoire to attack the cancer, leaving all the rest of the T cells, which we need for immunity against viruses, bacteria, but also we don't want them pouring out toxic cytokines when they're not actually addressing the cancer.
Yeah, fascinating. Well, thank you so much, Matteo. Really appreciate your education on this, and I'll look forward to seeing what happens next from here.
Well, thank you so much. Really appreciate the opportunity to discuss our data and our platform. So, Jonah, thank you very much for having me.